So, yeah, thanks very much for the opportunity to present on the European Lead Factory. This is it's another IMI-funded programme, which started in 2013 for 5 years, and it's another programme which has a very strong Dundee University component, who are my employers. So IMI, it's set up to fund, I guess, problems or gaps in translational drug discovery research. And the gap that the ELF was formed to address was that of providing high-throughput screening and hit discovery infrastructure to academics and SMEs throughout Europe. So we fit very much at an early stage of the value chain, the discovery of hit matter. And that can be as chemical probes to really validate the target as potentially interesting for therapeutic development, or as dedicated drug discovery programs. So it really is a large-scale experiment involving these four facets. So shared use of compounds. So there's 7 FPA partners who are part of this consortium, which is 30 partners in total. And they contribute compounds, proprietary compounds from their library to make up a larger library that can be screened. It involves crowdsourcing of innovative chemistry and biology, so it's not just a closed shop to the consortium partners, but it's open to anyone who's eligible to apply. There's an inbuilt mechanism for partnering with those FPA partners who are involved. And importantly, for a public-funded programme, there's a very strong emphasis on value generation. So the exploitation of IP out of this is essentially important to the process. It truly is, it's a pan-European collaboration. So I'm based up in Scotland, paid by Dundee University, but work near Glasgow. And there are academics, there are SMEs, chemistry SMEs, and there are the 7 pharma companies. There are 2 facets to the European Lead Factory. There's the joint European compound library, which is the compounds that we screen against the targets. And then there's the infrastructure in what we call the European Screening Centre that allows us to screen those compounds against public targets. The library is open to anyone in Europe, but it's also open to the 7 contributing companies. So they screen their own targets, and they screen their own targets in-house. In terms of the European Screening Centre, it really has all of the infrastructure that any major pharma has. It's based predominantly around legacy sites left over from the retraction from Europe from MSD in Scotland and in the Netherlands. And if we look at the compound collection, there's around about 300,000 compounds that are contributed by the FPA partners, roughly 50,000 each for the larger partners and 25,000 for the smaller partners. And a major goal of the programme is to reach out to new areas of chemical space to provide something very unique that's not been screened previously. So the concept is to build a new 200,000 collection over the lifetime of the project of new chemical matter that's out with the current scope, current chemical space. And that's a higher fraction sp3, more chiral centres. So the idea to try and develop designs that are more kind of natural product-like, I guess, whilst retaining the drug likeness profile. And there is a mechanism whereby chemists around Europe can contribute those ideas for monetary reward. But what I'd like to focus more on are the target contributions. This, again, I said, academics and SMEs who've got a molecular target that they've got a hypothesis for potential therapeutic regulation of, they can apply in a relatively straightforward application process. There are a number of requirements of the target owner, so they need to have a high-throughput screening-ready assay that can be developed onto the UHTS system. They need to be eligible, so they need to be an SME or an academic. And the applications are scored, they go through a review process. But once they gain access to the ELF, all of the all of the screening, all of the infrastructure, all of the reagents, etc., are covered within the screening centre, within ELF budgets. So basically, there is no cost, upfront cost for a target owner. It's not quite a free lunch. There are some responsibilities that come out the back end of that, and commercialisation of the output can trigger milestone payments. And one of the key principles is that after 3 years, if it's not being exploited commercially, it has to be published. The idea of compound sharing is unique within pharmaceutical company, certainly on this scale. If you're being cynical about it, you suggest that the companies might just open up the dusty old drawer with the rubbish in it and chuck that into this sort of library. Now we know that that's not the case. There's this article describing the makeup of the FPA core. This is the compound submitted by FPA. They certainly follow all of the principles of drug likeness, lead likeness, and it is very diverse. So there was quite a rigorous selection process which maintained the confidentiality of the structures within the individual libraries, but allowed deduplication. So we've got quite a diverse library of 300,000. And it fits very well with the properties of the drug likeness of other collections. The concept of building a very large infrastructure, a complex consortium, taking a very long time. This was actually put together quite quickly. Started in 2013, the selection of the FPA libraries, and then delivery to the logistics centre in Newhouse in Scotland. Took 6 months or so, and they were all plated and delivered to the 8 screening sites, by August 2013, and we were screening the first public target, around about October of that year. In terms of the development of the new library, the first delivery of new compounds was around about 6,000 compounds at the beginning of 2014, and that's now up to around about 60,000, 70,000 new chemical entities that have entered into the library. So they're very much on track to deliver that 200,000 towards the end of the programme. Just wanted to kind of touch on what a successful applicant to ELF can expect in terms of the running of a programme. It's very much centred around the programme owner, the academic or the SME, who has all of the experience and knowledge around the target. They should really drive a lot of the discussion around what it is we're trying to discover. And we form programme teams where we have a close dialogue with the target owner and the experts within the screening centre to develop a programme plan which will hopefully deliver us what we call a QHL, a qualified hit list. And this is the main deliverable of the ELF. And that's a list of 50 hit structures which that target owner has exclusivity on the development. So basically they have the IP on those compounds, the development. And that's one of the main things that goes back. Now in terms of getting to that qualified hit list, the confidential work takes place within the European Screening Centre. So we are the honest brokers of the library. And our plan, based on the programme plan, is to get to a preliminary hit list. And up until that point, actually, no one out with or within the consortium can see the structures within the database. We've got a very elaborate database which blinds us to the compound. So it's very data-driven in the selection of hits. That preliminary hit list needs to be validated, so we obviously don't want to contribute to the publication of dross in the literature for new chemical probes. So we spend a lot of time in biophysical approaches validating those hits. And then ultimately we deliver a list of compounds which we need to get clearance from the compound owners for. There could be any encumbrance on those compounds since they've been added to the library. There's a quick check. And then that list is then released and is legally fed back. There is also the opportunity to actually do something with those hits and expand them within the ELF budget. So we do some medicinal chemistry, functional assays, mechanism of action experiments to basically try and get as far along the hit-to-lead process as we can. And this is just an example of one of the projects. It's Chris Schofield from Oxford University. We screened 300,000 compounds against his targets, an enzyme-based target, biochemical assay. There was a deselection based on a related enzyme, which fed us just over 1,000 hits. Looking at potency and also running in parallel a selection of compounds for biophysical approaches in SPR, we identified really 60 binders which overlap with compounds giving good potency. And that was whilst the program owner can't see the structures, it's very much a dialogue of what it is they want to get out, based on the data that we're generating. And what to say about that is that in the 5 months since the qualified hit list was provided to Chris, we've got a very significant advance in that particular program. So there was compounds which are singletons within the individual company libraries, but once the libraries put together, created a series. So you got singleton rescue. We got resynthesized and reconfirmed the activity of the majority of the hits. The program owner, Chris, was really very engaged, and he put crystallography resource on it, and very quickly we were getting crystal structures of those compounds. We got significant improvements in potency down to picomolar, and most our favourable profiles of the compounds, tunable DMPK, etc. But most importantly, as part of this data package, is the delivery of, on his side, cellular efficacy, on-target cellular efficacy in extremely predictive models of the disease. So, and he's now looking at patent filing and possibly options with the FPAs, etc. So, and that's about it. So this is the full consortium makeup, and basically everyone's involved, and it's been quite a venture. And those are contact details if you're interested.
Thank you, Stuart. Thank you.